So, welcome to the second lecture. In the last lecture we talked about intercellular signaling. Now we look on another aspect, which is intracellular signaling. So, in the first lecture we just saw how signals can be transducted from one axon to a dendrite, so from one neuron to another neuron. And this transmission process can on one hand produce depolarization of the next cell, but it can do a lot of other things. So what we will do in this lecture is we will have a look on different types of receptors, on different receptor systems, and how they transmit the information they get to the cell. So we will look on ion channels. We had a brief look on ion channels in the first lecture already. We will look on so-called Gprotein coupled receptors, which are the receptors for neuropeptides as an example, and which are modulators of cells. We will look on receptor tyrosine kinases, which are important signals for growth factors and such things. And we will have a brief look also on steroid receptors, which play a role not only in sexual organs, but also in the brain. So if we compare these type of receptors, we have two different types of ion channels. One is the so-called ligand-gated ion channel. This is the version we already looked at. Beside this, we have transient receptor potential channels. So these are receptors which do not really act on activation of ligands in the classical sense, not of neurotransmitters, but they are basically reacting to other influences coming from the environment. Then we have the Gprotein coupled receptors, which in the end are neuromodulating function and have elicit a variety of different signalings. And then we have this receptor tyrosine kinases. You see they are mostly dimeric, and they are receptors for growth factors, induce signaling cascades, and have basically no fast signaling. And then we have the steroid receptors, which are basically receptors which directly transfer a signal to the nucleus and lead to transcription of new things. So let's start with the ion channels. And a slide you might remember. So in the first lecture, we had talked about the gradients of ions we have along the plasma membrane, which produce the membrane potential. So we have sodium and chloride outside the cell. We have a lot of potassium inside the cell. And due to this distribution, we have a large difference in charge. We have more positive charge outside the membrane, and we have more negative charge inside the membrane. And this is the membrane potential, and altering this membrane potential is basically the basis of the electric signal in the neurons. So if we depolarize these cells by sending sodium into the cell, we get the propagation of the electrical signal along the axon. This can be done by voltage-gated ion channels, which is basically the process we see when we get the signaling along the axon. It can happen on the postsynaptic membrane by ligand-gated ion channels. And this is basically the synaptic transmission, which we already looked at. The specificity of these ion channels, as we already said, comes from charge inside the pore. So basically, if the pore is basically negatively charged, then positively charged ions can go through, can go in, can pass through. But negatively charged ions will be repelled from the tunnel, and vice versa. And what we see is, if we look on these ions, which are important for signaling in the cell, we have sodium and calcium, which are on a similar level of size. So they fit mostly through similar types of channels, and frequently calcium channels can co-transport sodium, and in several cases sodium channels can co-transport calcium. There is much less calcium flowing because there is much less calcium around than sodium. And why this is, we come back a little bit later. Then we have the potassium, and you see the potassium is much larger than the sodium, so we can filter this. And we have the chloride, basically, which is again larger, and it has a different charge. So for this, we have the ligand-gated ion channels, which we already discussed last time, plus we have so-called transient receptor potential channels, and these are the channels which we need for sensing the environment. So basically these are receptors which sit in our fingertips, on our skin, where we can sense heat, where we can sense acids and so on. The signaling type is basically the same as for the ligand-gated ion channels. It's opening of a pore. And through opening of a pore, we get depolarization, action potentials, which then can lead through sensory neurons, through the dorsal horn of the spinal cord to the central nervous system. What these ion channels also can do is basically, if they are overreacting, then they can lead to a calcium overload, mitochondrial dysfunction, and basically a localized defunctionalization of the receptors of the cells. And that's what basically we know also when it happens when we get a burn, then tissue is damaged and so on. This is in part due to these channels, and that we don't feel so much in this place anymore afterwards. May also be caused by this. So this is not only working in the periphery. We have such transient receptor potential channels also in the brain, and some of them have some endogenous agonists as well. So you see them here. And maybe the one which most of you might heard of is the TRPV1, and if you not heard of, you might have experienced it. This is the one which gives you the sense of hot chili. So it's sensitive to capsaicin, and the capsaicin is basically the substance which makes a nice habanero feel hot. As we discussed in the first lecture shortly, such ion channels can be modulated. So the modulation of these ion channels can basically influence their function on different levels. It can function on the level of the opening probability. It can influence on the opening length and on the opening frequency. All these three parameters basically are important for the conductance. So if I open it more easily, if I open it more frequently, or I open it longer, then more channels can flow through. And if I restrict this, less channels can flow through. So basically, this modification of the receptors decides how much signal is produced on the same amount of neurotransmitter. So the presynaptic cell can send as much neurotransmitter as it wants. If there is no receptor, there will be no signal in the target cell. So this is very important. It's also important that these receptors can be completely desensitized. So if we overdo, if we treat the receptor too frequently, then it can be desensitized, and it can be also internalized, so taken away. And this is basically a process which is frequently seen in treatment of patients. It's also seen in addiction. It's called tolerance. So you need higher and higher doses of a drug to reach the same signaling intensity. On the other hand, of course, internalized receptors can be brought back to the membrane again and then can increase signaling. So as we said shortly before, calcium is a very special type of neurotransmitter. It's not a neurotransmitter which, like the other ions, basically just modulates the membrane potential. It's much more. It's not in a sufficient amount to really modulate membrane potential. But what it does, it plays a crucial central role in a number of secondary signaling, in the modulation of the cell itself. Yeah? So it can modify or activate calcium calmodulin. And calcium calmodulin is a very important enzyme which can on its own phosphorylate and activate a number of other enzymes. It can also activate protein kinase C, which is again a kinase, so an enzyme which puts the phosphorylation on other enzymes. And what we have here is basically calcium as an initial trigger of so-called kinase cascades. And these kinase cascades basically are important to amplify a signal. So we get from a single calcium, we can get or stimulation of different proteins, and these different proteins then can stimulate again a variety of proteins, and this spreads. It's like the snowball principle in economy. And that's why it's so extremely important to control calcium as an ion. So we have very specialized devices to control the presence of the calcium in the cytoplasm. So it can be taken up into internal stores like the endoplasmatic reticulum. It can be taken up by mitochondria, and it's strong transporters to get rid of it. So calcium is especially strongly controlled, although it looks like a normal ion from outside, but it plays a much more crucial role. This brings me to the next type of receptors. So this is the Gprotein coupled receptor. This type of receptor has 7 transmembrane domains. It frequently comes as dimers, can be heterodimers or homodimers, so consist of the same type twice or different types of receptors. There is no pore, so basically no ions fluxing through. The signal is transferred differently. The signal basically comes through the activation of the Gprotein. Gproteins are very frequent in the cell and involved in a number of different processes. There is one type of Gproteins which basically interacts with such receptors. And these Gproteins is basically consisting of 3 subunits, alpha, beta, and gamma. And in a resting state, this Gprotein is coupled to GDP, so guanidine diphosphate, and it's not active. And it's just closely sitting to the Gprotein coupled receptor, but it's not interacting. Now, if an agonist comes in, it binds to the extracellular part of the Gprotein coupled receptor. Again, we get a conformational shift which affects the structure of the entire 7 transmembrane domains of the Gprotein coupled receptor, opening a binding pocket for the Gprotein coupled, for the Gprotein. So basically the affinity to the Gprotein is increased. The Gprotein interacts with the receptor and is modified by itself in a way that the affinity for GDP is reduced and the affinity for GTP, so the guanidine 3-phosphate, is increased. by this process, the Gprotein is basically activated. And this activation of the Gprotein leads to a disruption. So the subunit alpha leaves the complex, and beta and gamma and alpha are separated. And they have different functions. So beta and gamma, in their form without alpha, they can directly interact with other proteins. Some of these proteins are involved in signaling, like ion channels, so they can interact with potassium channels, they can interact with calcium channels, they can interact with chloride channels, with sodium channels, and can basically either inhibit or facilitate these receptors depending on the type of beta and gamma subunit. So through this we get a fairly fast signaling. It's not as fast as ion channels, but it's pretty fast, and it can be lasting, and it influences fast signaling. The alpha subunit interacts with different targets, and the interesting thing here is that the alpha subunit itself has a phosphatase activity on the GTP. So basically, the alpha subunit can cleave the GTP, which is bound to GDP. And with removal of one phosphate from the GTP, the transformation to the GDP, the alpha subunit regains its affinity for beta and gamma, and this triplet comes together again, and basically either can interact with the Gprotein if the ligand is still bound, or it basically goes back to resting position. So this is what's called the Gprotein activation cycle, and this is a crucial thing for Gprotein coupled receptors. So as I said, the alpha subunit can interact with a number of different proteins, and we have a number of different alpha subunits doing that. So we have two types of alpha subunits, the G-alpha S and the G-alpha I, interacting with adenylyl cyclase. Adenylyl cyclase is an enzyme which transforms ATP, adenosine 3-phosphate, in cyclic AMP. And this cyclic AMP is a strong signaling molecule. G-alpha I basically stands for inhibitory, and the S stands for stimulatory. So you can easily read from that the G-alpha S facilitates the formation of CAMP, while G-alpha I inhibits the formation of CAMP. Some of these inhibitories alpha subunits are called G-alpha O, but this is just due to classification differences. So this cyclic AMP can activate a number of signals. It can also activate Cre, and therefore interact then directly with DNA in the nucleus and directly control gene expression, and that's the way how modulation can be done. The whole cell can be affected on the long run. Then we have this another very important group of G-alphas, and this is the G-alpha Q. The G-alpha Q basically activates phospholipase C, which basically splits phosphoinositol pyrophosphate into diacylglycerol, which is important to activate protein kinase C. And this protein kinase C is basically a starting point again for a kinase cascade. And it can also induce, through the production of IP3, the release of calcium from internal stores, especially from the endoplasmatic reticulum, and together with the diacylglycerol, this is important for the activation of protein kinase C, and this calcium released from the internal stores can do all the things which we just discussed for calcium in general. So there's very broad activation of a number of secondary signaling through these Gprotein coupled receptors. So they do a lot of modulating things. If we look on this cyclic AMP and the function of the cyclic AMP, one of the key functions is the activation of protein kinase A. And again, it's a protein kinase, which again is a starting point for a protein kinase cascade. So normally in the cell, the catalytic domain of protein kinase A, shown in blue, is bound to a regulatory subunit. If there is sufficient CAMP, cyclic AMP, in this case we need 4 of them, then the regulatory subunit is removed from the catalytic subunit. And the catalytic subunit now can start to phosphorylate proteins in 2 different types of amino acids, it's serine or threonine, by consuming ATP, energy from ATP, and it can phosphorylate. Now, it's important also to terminate such signals. Although the function is prolonged, you have to terminate such signaling because otherwise you would run into problems. So one of the enzymes which can be phosphorylated by the catalytic domain is so-called phosphodiesterase. And phosphodiesterase, upon activation, basically can cleave this diesterase bond and transform CAMP into AMP. And thereby basically stops the signaling because the catalytic domain then regains its affinity to the regulatory domain, because the regulatory domain is getting rid of the CAMP, and the reaction stops. If you get a long-lasting signaling, then the regulatory domain can be degraded and the catalytic domain remains active for a prolonged time, and that's something which is an important process during learning, but that's we see later on. Protein kinase C is activated through a different process, as we said. It's dependent on the G-alpha Q, and the G-alpha Q subunit basically activates phospholipase C, and this phospholipase C basically splits an integral membrane component. Phosphoinositol pyrophosphate is basically compartment building. It's the basic structure of the membrane, and it's split into deacylglycerol and IP3. So the deacylglycerol can activate the protein kinase C, but for this it's also needed calcium. And this calcium can be released, as we said before, from the internal stores like the endoplasmatic reticulum, where we have IP3 receptors, ranitidine receptors, which can release calcium. And with this, we get the functioning. And again, signaling. So the signaling can be terminated on the level of the protein kinases, because once they are activated, they would be independent of the Gprotein coupled receptor. But of course, the signaling through the receptor has to be terminated as well. So the neuropeptides which activate these receptors have to be cleaved and will be degraded. They will not be recycled. If you have classical transmitters activating Gprotein coupled receptors, you might remove them from the synaptic cleft by reuptake. In some cases, though, the signaling lasts too long for the cell. In this case, another important molecule which can interact with the Gprotein coupled receptor comes into play, and this is called beta-arrestin. It's more or less a molecule which can interact with Gprotein coupled receptors once they are activated. And this beta-arrestin then can recruit a kinase, which is called Gprotein coupled receptor kinase, which can basically modulate the Gprotein coupled receptor by phosphorylation, which then can lead to desensitization by internalization. So basically the receptor is retracted from the membrane. And by retracting the receptor from the membrane into a vesicle, there is still signaling. Yeah? Because what we have, we do not depend on an ion channel. Yeah? So what we have is, if the ligand is still bound to the receptor still is active, irrespective whether it's sitting in the membrane or in the vesicle. So if I remove an ion channel from the membrane, then basically I remove it from the gradient and there will be no flux anymore. If I remove a Gprotein coupled receptor, it still can be active. So what's necessary to stop the activity of this receptor is basically to pump protons into this vesicle, and through the acidification of the core of this vesicle, the ligand drops off. And only if the ligand drops off the receptor, then the receptor is no more stimulated. And then basically we have two ways the internalized receptor can go. So either it's basically recycled back to the membrane and reintegrated into the membrane and can be used for signaling again, or it's degraded. The beta-arrestin, beside this desensitization, can have also other functions. So it can trigger internal signaling as well, and this internal signaling can be quite different depending on the type of receptor you look at, on the type of beta-arrestin which is activated. And this signaling is again signaling which more or less goes through kinases and can affect gene expression. And it's quite interesting that this beta-arrestin pathway and the Gprotein pathway, they were seen as happening in parallel on Gprotein coupled receptors. And it was seen as something, one mechanism. Our new understanding is that this very much depends on the type of ligand, on the environment of the receptor, whether both are stimulated in a similar way or whether one of the two pathways is more prominently activated than the others. In this case, we would talk about biased agonists, and this is a development which is very, very important for drug development in terms of reducing unwanted side effects, because the effects we get from the activation of the Gprotein can be very different from the effects we get through the activation of the beta-arrestin. So if we can specifically activate one of the two pathways, we could get rid of a number of unwanted side effects under certain circumstances. So we have different signaling through a single receptor on quite a broad level. So we can have a direct signaling of the beta-gamma subunit of the Gprotein interacting with ion channels. We can have the alpha subunit interacting with signaling cascades, might be cyclic AMP, might be protein kinases, and we have the beta-arrestin signaling. So a single type of receptor can produce a number of different signals, and this depends very much on the environment, on the ligand, and in different cells we have the same type of receptor interacting with different Gproteins. So it's not necessarily that a Gprotein coupled receptor produces the same effect in each type of cells. So that can vary. Okay, let's go to the next class. Basically, receptor tyrosine kinases. These receptor tyrosine kinases are receptors of growth hormones, of growth factors. They are more or less signaling only in a metabotropic way, so by influencing some relay proteins, some proteins which might stimulate other kinases again. So the signaling molecules here is frequently molecules which are important in sensing states of the brain. Yeah, it's growth factors which might derive from other neurons that can be also signaling backwards from the target neuron to the original neuron. That might be signaling which derives from glial cells. So it's information not so much involved in direct neurotransmission, but it's very important in priming the state and preconditioning the neurons. And again, we have signaling cascades, and one of these signaling cascades we see here. So in the end, this signaling cascade can influence kinases which are important for gene transcription. So we can have signaling back to the nucleus and modification, cell proliferation, apoptosis, and such things. So through signaling, through growth factors in the brain, it can happen that neurons form new axons, that they form new synapses, or that they also die. So it's a process which is quite important in a number of neuropathological processes, in neuronal degenerative diseases, and it was discussed in Alzheimer's and so on. And treatment is difficult because it can produce a number of effects and it's not easy to control. So basically these are processes which are important for the neuron to be predisposed for signaling. And the other class which we have to look at is so-called nuclear receptors, steroid receptors. So these receptors basically have no direct metabotropic function. So they are not directly involved in neurotransmission, but they regulate gene expression. So basically what we have here is signaling which goes directly to the nucleus and directly involves mRNA synthesis. In this case, what we see here, it's a testosterone receptor, so a steroid receptor, and this plays a lot of role in the transition of the brain from a premature to a mature brain. So these sexual hormones have a strong influence on the brain, and this influence becomes strongly visible also to the environment during a process called puberty. Yeah. So in puberty, when steroids, sexual hormones rise, we have a strong impact on brain function. We have a reorganization of some parts of the brain, changes in functions, and that's important for the juvenile brain to switch into a mature mode. During this period, of course, there is some issues sometimes due to this reconstruction. Yeah? And mature brains sometimes have problems to understand brains under reconstruction. So these nuclear receptors, basically the hormone itself has a structure where it can pass through membranes. So these hormones themselves do not need a transporter, they do not need a receptor in the membrane, although some of these receptors can sit in the membrane. And in recent years it was also shown that they can have some metabolic influence, not only translational, but the main issue is the transport into the nucleus and RNA synthesis, which leads to to protein synthesis, which then changes the cell function. As I said, in recent years, also membrane-sitting receptors for steroid hormones were found, and they are not these classical steroid receptors, but they function more metabotropic, as they are Gprotein coupled receptors. So the steroid hormones have also other receptors. Now we have seen different classes of receptors, different signaling pathways, and we looked on them quite isolated. But of course in a single cell this can integrate very much. Yeah? So considering that especially classical transmitters might have different types of receptors. So we find for glutamate, for GABA, so on, we find ionotropic receptors, and we find Gprotein coupled receptors. So we can basically get a signal through an ion channel. This ion channel signaling might be modulated through signaling through Gprotein coupled receptors. The cell itself might react differently due to the fact that Gprotein coupled receptor was activated, and we get a stronger polarization of the membrane, so it would need more depolarization to in the end generate a action potential. This integrating of signals through different receptors, and this includes also the receptors for growth factors and so on, is also a crucial point in regulating cell survival, in regulating cell proliferation. Well, you might have heard that mature neurons in the end do not proliferate, but we have a small pool of progenitor cells in the brain still active, also in the adult brain, and of course it also regulates the development of the brain during ontogeny. And these complex things interacting on each other makes it so tricky to really understand what the modulation on a single level would mean, because modulating on a single level might influence the reaction of different other signals, and that's not such an easy issue. So we can have influence on the fast signaling, we can have influence on the neurobiochemistry of the cells through these different receptors. So we might have phosphorylation of receptors, changing affinities of receptors so that they have different affinities to their ligand. So if we have an increase in affinity to the ligand, then less neurotransmitter would induce the full signal. It might influence existing proteins through phosphorylation. Dephosphorylation might influence the activity of different proteins, and it might influence the synthesis of new proteins. So we can have new proteins integrated into the cell, integrated into some parts. And this is things which are crucial for the normal brain function. The brain is a very, very dynamic thing, and it's not rigid. It's learning. It's modulating itself every second, every day. And to get these things, we need a very complex system. We need to get the best out of the cells. And we have, for each neuron, we have 10 to the 4th synapses. In some cases, 10 to the 5th. And each of these synapses can be modulated differently. So it's quite a complex thing, not only in the network aspect, but also in the signaling network. Yeah. So this can be really complex, and it will be your task to simulate that on a computer. Thank you for your attention.